Okay, so let's join. So welcome to our lecture again. Yeah, to this today we have a special guest, which is Fabian Ross, that you see on the other window. And as you remember, we before Christmas we introduced theoretical physics concepts that explain how order emerges in non-equilibrium systems. And then last week I started introducing some basic tools from data science, namely what are the tools that we can use to computationally deal with large amounts of data. And today, so I need some special help from somebody who's a real expert, which is Fabian. Fabian is a trained physicist and did a PhD in mathematical biology, then did a postdoc in my group at the Max Planck Institute in Dresden. And now he's a professional data scientist and bioinformatician at the Center for Regenerative Therapies in Dresden. And so he's talking actually about something that he's doing for his professional job, which is very nice. And over to you, Fabian. Thanks a lot for sharing your expertise.
Yeah, thanks for having me here, for giving me this opportunity to give a lecture on exploring high-dimensional data. So I think Stefan introduced me very well. Thank you very much, and also made the point of where we are in the course. So I just wanted to ask you, to say that whenever you want to ask something, I think during the course you just like unmuted yourself and ask the question. So that's totally fine to me. Or also type something in the chat and I try to answer. And then like if you're not saying anything, please mute yourself to reduce noise. All right. So okay, I'm going to start right away. So today will be about exploring high-dimensional data, right? And as many of you know, with high-dimensional and in high dimensions, strange things can happen. So if you, for instance, look at this thing here, what you see here is a three-dimensional projection of a four-dimensional object rotating, and this looks very funny, right? And I mean, like 4 dimensions is where we kind of still can kind of get a glimpse, but what if it gets to thousands or millions of dimensions? What do we do with these kind of data? So before I start, let me give you a brief structure of the course today, of the lecture.
You might have to click into the window.
Yes. No, I actually want to share
okay.
my iPad screen. Yes, sorry for the delay. All right, so I basically start by introducing what I mean by high-dimensional data. I will then cover dimensionality reduction methods. And also talk a bit like what's the idea of reducing dimensions. And third, if we still have time, I'll talk a little bit about clustering, which we kind of can think of as a discrete form of dimensionality reduction, I'd say. So what do I mean by high-dimensional data? Well, let's start with an object that I call a data matrix. X, which is of shape n by m. And maybe you remember from last week, tidy data format. So this matrix should be in a tidy format already. So in the rows, we have n samples. And in the columns, we have m measurements or observables. So what do I mean by this? So an example could be what you measure here is, for instance, you take yourself, right, and you measure, for instance, the length of your thumb and the length of your index finger and the length of your middle finger and so on. You do the same thing on with the other hand. You measure your foot size, your body height, maybe the heartbeat rate, and so on, and you end up gathering more and more observables. And that's M observables, and this gives us the dimensionality of our data. And now you can do this with yourself, and you can do this, you can ask your friends to do it with them and so on. And then you can maybe do it with all people in Asia or all over the world. And this gives you the number of samples that you have, right? Now, how do you treat these kind of data, right? If we go one step back and think about how can we visualize, and Steffen uploaded some slides on that, and maybe we'll also talk on that a little bit. But what can we actually visualize easily? So if you would have one-dimensional data, right, that's rather easy, right? You can just plot your data on a one-dimensional line, for instance. And also for 2 dimensions, I think this is pretty straightforward, right? So if this would be the entries of our data matrix x1, x2, we can just plot them as a scatter plot, right? And we could, for instance, see whether they are correlated or not. But as soon as we are like in 3 dimensions or larger, like 3 dimensions, we can still handle. We can do a 3-dimensional plot maybe, but this is still hard to quantify. This gets non-trivial, right? And so we need techniques to think about how to actually look at these data and how to analyze these data. So the aim of dimensionality reduction then, I'd say, is to reduce the number of observables while keeping relevant information. And what this relevant information is, right? This is something you have to decide by the problem at hand. Now, before I go into talking about how you can reduce dimensionality, I wanted to show you some examples. So I'll share some slides for this. And so I think as many physicists are in the audience, one example I wanted to share something that I think many of you are already know, which is where many of you know that a low number of observables actually suffices to describe a system very well, and that's in the realm of statistical physics. And you saw a lot of examples from Steffen by that, but even in a rather easy example like an ferromagnetic isoing model, right? You know that there's many spins in this system, right? And imagine you could measure all these spins. You have a very high dimensional representation of the state of the system then. But many of these spins will be correlated, and we know from statistical physics that actually you can very well describe the macroscopic state of such a system with just the magnetization. Now, what will happen if we come from the other side, from the data science perspective? And that's a paper here in arXiv by Sebastian Wetzel. And what he did was he did a simulation of a ferromagnetic Ising model on a 28 by 28 lattice at different temperatures. And so 28 by 28 gives you 784 spins, right? And he recorded the state of all these spins in 50,000 different samples at different temperatures. And now you have a data matrix with 784 dimensions, right? And then you can start to say, okay, if I just would have this matrix and I would know nothing about the system, how can I look for order in this system? And how can I reduce dimension? And one of these techniques is called principal components analysis. And I'll explain later what this actually is. But if you do this, you get a result like this. The first principal component, which is a one-dimensional representation of this high-dimensional data matrix, perfectly scales with the magnetization. So in this way, you would actually be able to kind of discover an order parameter without knowing anything about the system by having detailed microscopic data of the system. So this is rather an artificial example, right? Because we know very well about how an Ising model is behaved. But what about systems where we don't really know how they work? For instance, humans. So this is a paper from Nature in 2008. Last author was Bustamante. And what they did was they quantified human DNA. So they took 3,000 European humans. And now you can imagine that the DNA is a long string of characters, right? And to a large part, this is conserved from one human to another human. So the DNA will be pretty much the same. But at some positions, we have variations, right? The DNA from one human to another is not exactly the same. And at 500,000 positions, the authors quantified the DNA state. And so this gives you a data set with a dimension of 500,000 approximately for 3,000 humans. And then they did a dimensionality reduction. And again, they did a principal components analysis. And this is a two-dimensional representation of this high-dimensional data set. And what I want to ask you now is to look at the picture, and you don't yet have to understand what the principal components mean, but I want to ask you to type in the chat if you can what you see in this picture.
It's a very difficult or very shy audience. What does it look like? To me, it looks like an elephant.
It is a scatter plot, yes, and we see some clusters, yes.
But there's also some sensible answers.
Yes, and it's actually no, I don't see my mouse anymore.
I think Adolfo got it right.
Yes, Adolfo got it right. It's already the second answer. It resembles a map of Europe, and that's exactly what the title of the paper was. Genes mirror geography within Europe, right? So this was, I think, is quite amazing. So you just record the DNA of humans, right? And basically this gives you a map of Europe approximately. It kind of makes sense, right? That genes are well, somewhat remain local. So here's another example. That's a very, I'd say, maybe unphysical example, but I think it works nicely. These kind of examples using images work nicely to explain dimensionality reduction techniques. So this is a fashion data set called Fashion MNIST, provided by Zalando Research. And again, you can, these images are on a 28 by 28 lattice in a grayscale. So you can record the intensity of each pixel, and you end up with having 784 pixels of 60,000 pictures. And there's different kinds of pictures, like there's shirts and tops and trousers. There's also shoes, I think, in there. And then you can do a dimensionality reduction. And here this is an example for a UMAP, a uniform manifold approximation. I'll explain later what this is. It's a nonlinear dimensionality reduction technique. And again, you can see a number of things. By just taking this as vectors, right, and projecting them in 2 dimensions, you see that, for instance, I think this up here, which is trousers, forms one cluster, which is very different, for instance, from a cluster which consists of sandals and sneakers and ankle boots. So this is the kind of shoes cluster. You have bags here, which seem to have some similarity to tops at some point in a certain way. So this is again dimensionality reduction, right? And you can see how you can use it to group together different kinds of things. In this case, just pictures. What you also see here is that these things cluster. So there's a discrete kind of order, and I'll talk about clustering also again in the second part of the lecture. Now I'll go into dimensionality reduction and how it works, and I'll start with a linear dimensionality reduction. There's a number, so this is just a linear transformation of your data. There's a number of techniques around. For instance, principal components analysis, I'll explain this, and there's other techniques like non-negative matrix factorization or independent component analysis or factor analysis. They are kind of similar in that they are linear, but I only have time to cover one of them, which is the easiest one, which is PCA. For that, I'll again share my iPad. All right, what's principal component analysis? I'll first sketch the idea. So for getting the idea, it's sufficient to go into 2 dimensions, and I'll make a plot, a scatter plot of 2-dimensional data, where let's assume our data looks a bit like this. Right, it's essentially very well correlated, and from looking at this now, if you would give me x1, right, and I would know x1, I could very well predict x2 because I see that these 2 observables are very well correlated. And then PCA is a way to formalize this, and the first principal component is designed in a way that it points in the direction of highest variability of the data. And in this example, this would be this direction. And this would be principal component 1. And then the next principal component has to be orthogonal to PC1, and again point in the direction of maximum variability of the data. And there's only one direction left here, which is the orthogonal direction here, and that's PC2, principal component 2. So then you just change the basis of your data to the directions of the principal components, and then you end up with your representation of your data like this. Where now in these directions, your data would be uncorrelated, and you would have most of the variability in this direction, right? So the aim will be to find a transformation such that the components are uncorrelated and that they are ordered by the explained variability. Now, how does the math of this look like? Sorry. Mathematically, we would start with an eigendecomposition of the covariance matrix. The covariance matrix is an object which we get from, or at least this is proportional to this object. It's the data matrix transformed times the data matrix itself. And you will see that this is a matrix of the shape m by m. So it has the dimensionality of our data. And if you do the computations, you will see that the diagonal elements of this object are proportional to the variability of our data in this direction, and the off-diagonal elements are proportional to the covariance of components. And then we do an eigendecomposition. And from this, we get m eigenvectors lambda, m eigenvalues lambda, and m eigenvectors, and I group them together in a matrix W. So the columns of this matrix are eigenvectors. Of the covariance matrix. And importantly, what we do here, and we can always do this, is we sort them by the magnitude of the eigenvalues lambda I. And this ensures that the first principal component points in the direction of highest variability. Now, W is just the new basis for our data, and then we can transform our data. So like this, the transformed data will look like this. We take the data matrix X times W. So in the language of principal components, this is what's called loadings, or also the rotation. And this, the transformed data, is called scores. Now I leave it to you as an exercise to show that the transformed data matrix is a diagonal matrix, which basically means in this transformed space, the components are uncorrelated. Now the data matrix, again here, is of shape n by m. So the dimensionality of this is just the same as the dimensionality of our original data. So how does this help in reducing the dimensions? Well, there the idea is, because the eigenvectors are sorted by the magnitude of lambda I, that we can truncate it. And if we define WR, which is an n by R matrix, as the first R columns of W, then we can get a projection in lower dimensions of our data just by using RW, WR. And this object will be of shape n by R. And if we, for instance, select R equals 2, we have a two-dimensional representation of our data. Of course, this throws away some information, but most of the variability of our data is captured. Yes, so the take home is that principal component analysis is a linear transformation of the data such that the components are uncorrelated and ordered by the amount of explained variability. And what I want to do next is I want to show you how you can actually do this in R, which is quite easy, and you don't actually need to know the math for this. So let me.
Excuse me.
Yes.
Could you repeat, how do we decide at which point to truncate W, and what exactly do we mean by sorting by magnitude of the eigenvalues lambda?
Okay. If you this is the covariance matrix, right? So essentially, if I look at this, sorry, you didn't. Do you see my mouse?
Yes.
Yes. So lambda 1 will be proportional to the variability in this direction. Okay.
Yes.
And lambda 2 to the variability in this direction. Okay. And so this sorting by the eigenvalues ensures that the first component, the first principal component, will point in this direction of highest variability of the data, right?
Mm-hmm. Yes.
So this is what the sorting means.
Okay, so in this case, lambda 1 is that the reference that we sort through, that we get W from, because lambda 1 is proportional to the PC one direction.
Lambda 1 is the eigenvalue, right? That corresponds to the eigenvector, which is in this direction.
Yes, yes.
And it is actually, and if you do kind of if you look at this, at the transformed covariance matrix, right, you will see that in the diagonal elements of this, you just have the lambdas, right? So it's that's the variability, the variance in this direction.
Okay. Yes. Thank you.
Exactly. So was this all? I
Yes,
really
that was.
remember there was another question. You also asked how to truncate, how to decide where we truncate. This is up to us. You can kind of what I will show you in a moment is that you can decide that you want to keep like 90% of the variability of your data, and this would give you a threshold of where you stop, for instance. But this threshold again is arbitrary, so you can also just one usual decision that you do is if you want to visualize your data, you truncate at 2 because it's easy to visualize. You might also take this as an initial step of dimensionality reduction to maybe just like going from 500,000 to 50 or something, and then do a nonlinear transformation. But essentially, like in my everyday work, this is it's often an arbitrary choice, and you in the end have to see whether it works or not, whether it's sensible. So that's a tough question, and there's also some research still on that.
Okay.
All right, so I prepared a document, a markdown document, a notebook in RStudio, and I think Steffen briefly introduced RStudio to you last week. You can also get the code on from this URL, and it's also by now on the course homepage.
And also the code from the last lecture. I forgot to mention that. It's also on GitHub in the same folder.
Yeah, exactly. And then, what I wanted to show you before is that, like, I want to just show you as an example one of these famous data sets you might have heard of. It's a data set where botanist and statistician Fisher collected flowers from a flower called iris, and there's different species: Iris virginica, Iris setosa, Iris versicolor, and these have different leaves, the petal and the sepal. And he measured the sepal length and the sepal width, and the petal length and the petal width for all these species. And it's a data set that you can easily load in R. That's why I used it. And it's 4-dimensional. So here you see the data. It's the length, width, and width for one species, and then the other species follow somewhere in the data. And you have this 4 numbers. Now you can try to visualize this in a, for instance, pairwise scatter plot, plotting the length of the sepal against the sepal width and so on. And here this is color coded by species, and you already see some separation. but it's hard to choose, like in this 4-dimensional plot, what to actually look at. And so you can perform a principal component analysis on this 4-dimensional data set. And in R, this is just a one-liner. So the principal components analysis is done by prcomp. And here I plug in the 4-dimensional data set. And then you get all the things out that we just talked about. You get this transformation matrix W called rotation here, where now you see that the direction of principal component 1 in this 4-dimensional space, right? It's a vector. You see, you can look at the transformed data, which is again 4-dimensional at first. Here you can look at the explained variability or explained variance. And this is maybe for the question of where to cut off. So that the explained variance, here you can see the cumulative proportion of this. And with the first two principal components, you explain 95 or 96% of the variability of your data. And you might say, okay, that's enough. With in PC4, from PC3 to PC4, you hardly add something. So the 4th dimension of the data set is probably very correlated to the first 3. There's not much information left. And then this is how the data looks now in the direction of principal components 1 and 2. And what you can observe is that cross principal components 1, that's the main direction which kind of discriminates between the species. You can also see that Setosa is very different from versicolor and virginica, and here it's hard to actually draw a boundary, but just based on the measurements. This is what's called a scree plot, and this is just again a visualization of the variance explained by each principal component. Now I wanted to also show you another data set where actually principal component analysis has a hard job to work. So this is again an image data set where images were collected of different objects and they are rotated, right? And now you can again do a vector representation of these images, do a principal components analysis, and this is what the picture looks like, and what the plot looks like now. And the colors now represent different objects, and the dots of the same color are then pictures of the objects at different angles. And you can see that if you do this dimensionality reduction, you can see some structure, right? But it's not really, doesn't really work good in discriminating the different objects. And you would expect that maybe you can reveal this discrete kind of order, the different kinds of objects. Or that's maybe what you want by doing a dimensionality reduction. Here's another example that I made up, which is a dimensionality reduction of a Swiss roll. So what's a Swiss roll? Actually, a Swiss roll with a hole. So what I did here was to say, okay, I randomly place points on a 2-dimensional surface, but there's a hole in the middle. And then I wrap this up in 3D like this. I transform it like this. Okay, it's this roll, and it's a suite actually in Switzerland. That's why it's called a Swiss roll. And there's, you can still see this hole in the middle. Now, what will happen if you do a principal components analysis of this? The example is shown here. And this is the same data shown in principal component space. Actually in 3D. And so what you can see is that not much happened, right? It's pretty much the same as before, it just stayed a principal component. And the reason for this is that what we do is a linear transformation of the data, right? But what I did before was to take 2-dimensional data and non-linearly project it into 3D, or use the nonlinear function. And so projecting this back to 2D in a way cannot work with a linear dimensionality reduction technique, and that's why nonlinear dimensionality reduction techniques are also very useful. So this leads me to the second part of dimensionality reduction, which is about nonlinear dimensionality reduction techniques. And as it's usually the case with nonlinear things, there's many of them. And then actually about 1 year ago, I had a look at Wikipedia at the a list of some prominent algorithms for dimensionality reduction, and there were 29. I'm sure now there's more, and this list doesn't claim to be complete, right? So this makes very clear that depending on what you want, there's different things that you can do. Today, I will concentrate on uniform manifold approximation, a rather recent one, introduced, I think, in 2018, so 3 years ago. And it's used a lot in single cell sequencing data, in single cell genomics nowadays. So what is this technique about? So don't be scared. There's a lot of mathematics on this page, and you don't have to understand all of that right away. I also won't have the time to fully explain this in detail, but I will give you an intuition of what this does. The basic idea is that the basic idea is that we have a high dimensional data set, but we hypothesize that there's a low dimensional manifold. So think of for instance, there could be a line in a 2-dimensional manifold, right? And then we want to discover this low dimensional manifold. So there's a couple of assumptions that are made in UMAP. One, that the data is distributed uniformly on a Riemannian manifold, that the Riemannian metric is locally constant, and that the manifold is locally connected. And what UMAP then gives you is it models this manifold with a fuzzy topological structure, and it finds a low dimensional projection of the data that has a very similar fuzzy topological structure to your original data. So what does all this mean? Let's start by, I will start by introducing very briefly introducing an important idea from topological data analysis, because this algorithm this idea is heavily based on topological data analysis, and this is simplices. A zero simplex is a point, a one simplex is a connection of two zero simplices, which is a line. Then there's a two simplex, which is a triangle, a three simplex, which is a tetrahedron. And you can see that you can easily extend this concept to higher dimensions. So you can introduce a 4 simplex, 5 simplex, and so on. And these objects are rather easy to construct. And a nice thing that you can do is you can glue together such objects, such simplices, and by gluing them together, you can basically approximate manifolds. So you might all have seen already some point where you triangulate a surface, right? And this is nothing than approximating this manifold with simplices. Now let's look at this example, which I took from the UMAP homepage actually, where you can see points which are uniformly sampled, actually equidistantly sampled, I think, on a, not equidistant, but uniformly sampled on a line in a 2-dimensional space, right? So we have a 1-dimensional manifold in a 2-dimensional space. And so this could be our data points that were measured, right? And we will be interested in finding the topological structure of this manifold. So what has been done here is to draw some spheres around these dots. And the union of all these spheres, I mean, all these spheres are sets, and the union of all this is what's called an open cover. An open cover is just the thing that is a collection of sets that span the whole manifold, right? So the line that we see here is inside this open cover. And now you can do some heavy topological mathematics, and you can then prove that if you now construct simplices by all groups of sets that that have a non-empty intersection, which in this case basically would mean you construct a one simplex here, line here, and a one simplex here, and so on. I think you get the picture. Then if you do this, and this would look like, then this structure, this graph-like structure that you get out of this captures the topology of the manifold. The information is encoded in there. And I think in this two-dimensional example, this is quite intuitive, that the connection of all these points tells you about, tells you that this is a connected line. And there's no loops, for instance. So that's pretty cool. And now we would like to use this concept to learn something about our high-dimensional data.
Could I ask something? Is the criterion for the connection always like the shortest distance between the points? Because I mean, here I could see it, but if I have more complex structure.
Exactly. I'm actually coming to this point in a second. In this case, it was a bit like, okay, let's just guess the size of the sphere, make it a good guess, such that it works, right? But if you have high-dimensional data, what would be the size of this sphere? That would be super unclear. So I'll come back to this question in a moment. It's also connected to a situation like this, right? Because our points might not actually be uniformly distributed, okay? So if we would do the same thing here, drawing these circles and construct the simplices out of that, we would actually get disconnected parts, okay? And this is not what we would intuitively like to get, right? Because here we saw this line. Now, and this is also, this kind of breaks the assumption of UMAP, which is the uniform distribution of data points on the manifold. So how can we circumvent this, or how can we solve this problem? We can kind of turn the problem upside down and say, no, these data points are uniformly distributed. We see different distances if we apply a constant metric. So this has to be wrong. The metric is not constant. Actually, we can try to construct a metric like this. And this comes back to your question. We might say that the distance, in this case, to the fifth neighbor should always, on average, give us a distance of 5, right? Such that we say distances between neighboring data points should, on average, be 1. And this would mean that a distance of 5, for instance, for this data point would be rather large, and a distance of 5 for this data point would be rather small. So we define we now use nearest neighbors, k nearest neighbors, to define a local metric. And we then turn this local metric, again, and I'm skipping a lot of details which are important here, into a construction of simplices, into a construction of graphs where the edges now are weighted. And the reason that they are weighted now is that the distances between points are different, and also that we actually have 2 measures of distance. One using the local metric from here to there, and one using the local metric from here to there, because they might not be the same. But if you properly do the math behind all that, you can show that this construction now captures essential parts of what's now called the fuzzy topology of this manifold. Okay, so we have a graph representation now that captures topological structure of the manifold we're interested in. How can we do that? How can we use this now to find a low-dimensional representation? Well, the idea is that now you start, you have this graph defined, and now you start with a low dimensional representation. You just place your points randomly, basically, and then you construct a similar graph representation of your low dimensional representation, and then you shuffle your points around until the topology, the graph representations kind of match. So yes, you have a graph representation of your high dimensional data, of your low dimensional data. You interpret the edge weights as probabilities, and then you minimize the cross entropy given here, where basically the important thing is that you try to match the edge weights in your high dimensional representation with the edge weights in the low dimensional representation. Like when this ratio gets 1, the term vanishes, and the same happens over here. Now, how does this work in practice? Like, if you want to read more about this, there's a nice homepage. I just wanted to show an animation here, which is called Understanding UMAP, where, for instance, here you have an example in 3D. You have dots which run out from a center in a star-like fashion, right? And that's a 3D represent. This is a 3-dimensional data. And, or actually it's 10-dimensional data in this case, I think, and this is a 3-dimensional projection maybe. And now if you run UMAP, these dots are shuffled around randomly, and you stop when you have a topological representation, which is very similar. And in this case, this is also this star-shaped pattern, but now in 2 dimensions. And if you want, there's many more examples here, and you can play around with this. You can, of course, do the same thing in R. I don't know, like, I know this was a lot of information, and I didn't go into the details. Are there questions about UMAP at this point? Otherwise, I just show you how to do this in R. It's essentially, again, a one-liner where you have a function UMAP. And what I feed in here is the Swiss roll that I created previously. Okay, and then I just I do the dimensionality reduction here with a one-liner. Then I convert the whole thing in a data table object to have a nice plotting. And this is what the result looks like. Now in UMAP coordinate, the Swiss roll was kind of unrolled, but it heavily lost its shape. But what you can see is that the topology is preserved, right? So the hole in the center is still there. I also wanted to show you a UMAP representation of the coil 20 data that I showed you above. So remember, this was the data set where we had pictures which are rotated, and where the PCA didn't look very informative. Now, if we do the same thing with a UMAP and we feed in these pictures, this is what we get. So most of the images nicely separate, right? Different colors mean different objects, and they nicely separate. And what the UMAP also gives us is this is the topology of this data, in the sense of that we can see that this is a rotation, right? So neighboring pictures are similar, and you rotate the object until you come back to the other side. Okay, I think, yes, that's it for dimensionality reduction. Just one example for linear dimensionality reduction, which was principal components, and one for nonlinear topology preserving dimensionality reduction, which was UMAP. And I now wanted to talk a little bit about clustering. So you can, as you already saw in some of the reduced dimensionality, in some of the representations of the data in reduced dimensions, clusters can appear in your data, which essentially mean you have qualitatively different objects in your data, different samples, and sometimes and it's good to have a way to group different kinds of different classes of objects together. And clustering is nothing which is, there is no one definition of clustering. The idea is to find clusters such that the data within clusters are similar, while data from different clusters are dissimilar. But what you define as similarity or dissimilarity, that's basically up to you. And so a lot of different clustering algorithms exist. And I just wanted to introduce 3 of them today. And you use this a lot in, or at least I use this a lot in trying to understand high dimensional data. I'm just going to share my iPad again. And I wanted to show you first, I want to give you 3 examples. And the first one would be hierarchical clustering, where here I just plotted some data in 2-dimensional space to illustrate the concept of clustering. And what you need for hierarchical clustering is, first of all, a metric which defines distance between your objects that you have here. And this could just be, for instance, the Euclidean metric or Manhattan metric or a correlation metric. You're basically free to choose. And then what you do is you group together the 2 objects which are closest together by your metric. So with the Euclidean metric for this example, this would be those 2 guys, right? So what you then create is a kind of tree where okay, I said we group together those 2 guys. So that's the first thing to do. And now next we need another thing which is called the linkage criterion. And this tells you how to now treat these clusters, because now you need a way to get the distance between the clusters. And this could, for instance, be the minimum distance. So this would mean if you want to now get the distance between this cluster and this object, you would take the distance between B and D and C and D and take the minimum distance. Or you could also, for instance, use the centroid. There's many of the clusters, there's many choices, right? Now, if we kind of go on, we maybe take the centroid here, then the next cluster would probably be a grouping of D and E. Then I'd say I would add those 3 in one cluster. Then group all these, B, C, D, E, and F, until finally we arrive at an object which contains all the elements. And now if we want to define clusters in our data, we need to define a precision threshold along this axis. And we could, for instance, put it here. And then this would cut the tree in separate subtrees, and we would have a subtree here, a subtree here, and a subtree here, and then this would be our 3 different clusters. So we would have a cluster 1, cluster 2, and a cluster 3. And if we increase the precision like up here, then we would get more and more clusters. And as this basically just needs some distance computations, this is rather easily done in also in high dimensional data. Next, I wanted to show you another important one, which is a centroid-based clustering called k-means clustering, and one comes across this quite often as well when working with high-dimensional data. So here in this case, you pre-specify the number of clusters k. So that's what you have to start with. How many clusters do you want to get? You might try out different k's in practice. And then you partition your data such that the squared distance to the cluster mean position, so that would be the centroid, is minimal. So how would this look like? Actually, maybe let's look at the final result first, where kind of here you see an algorithm converging, but what you try to do is to do a grouping of the data such that, oh no, it doesn't stop there. Sorry, so how do you get this partitioning of your data? One way would be the Lloyd algorithm. You start with random centers that you place somewhere. So in this case, this is an example taken from Wikipedia. There's a center here, a center there, and a center there. And then for each of your data points, you assign the data point to the cluster where the center is nearest, right? So the yellow center is here, so everything here would be assigned to the yellow cluster. Now, once you did this, you update the centers of your clusters. Now you take the centroid of this yellow cluster, for instance, and the yellow center ends up here. And now you do a new assignment of your data points to the nearest center. In this case, all the yellow ones up here would be assigned to the blue center, actually. So they are assigned to the blue, and then you iteratively go on and do this, go on and go on and go on until this algorithm converges and you end up having 3 clusters defined by this criterion. Of course, there's other more complicated, more sophisticated algorithms, but this approximates the solution. A third algorithm that I wanted to show you, and I want to show you this because I work with it every day, comes from social systems, which is a graph-based clustering. When you start with your high-dimensional data, you first need a graph representation of your data. And we already saw this in the UMAP example. You could, for instance, do a k-nearest neighbor network on your data. And then you partition your data such that the modularity Q is optimized. Modularity in this case is defined as this object where Eij are the fraction of links between cluster I and J, right? And what you try to do then in this object is that the links between cluster I and I, which is the internal links, it's this EII, right? It's the links inside a cluster. You try to maximize this, and in this AI term, you have the IJ from I to another cluster. So this is links out of the cluster. You minimize this, right? So you have very clusters means a cluster in this case means that it's parts of your graph which are densely connected inside, but only sparsely connected to the outside. There's a particular algorithm which is used a lot nowadays, which is the Lovong algorithm. So how do you solve this? Because this is not, you can't solve this brute force. You start with a network like this, for instance. This would be your nearest neighbor graph that you created from your data, and then you do copy attempts. You start with node 0. So all these now, you start with all these being separate clusters, and then you copy cluster identity 0 to neighboring nodes, and you again compute the modularity. And if the modularity is increased, you accept this copy step, and if it's decreased, you reject the copy step, and you do so until you arrive at a steady state like this, where copy attempts don't increase your modularity anymore. Would be in this case something like that, where you end up with 4 clusters, and then you do an aggregation. And this means now that you define a new graph representation of your data, where this cluster here, the blue cluster, is now a single node. It gets 4 internal connections because there's 1, 2, 3, 4 internal connections, and there is 4 connections to cluster green. 1, 2, 3, 4, and 1 connection to the light blue cluster. And so you aggregate this, you do the copy attempt again, and so on until you finally arrive at a picture where you at a state where you can't optimize modularity anymore. And this is how the algorithm works to solve the problem of modularity optimization. And this algorithm works very well with many kinds of data. Again, for instance, it's used a lot to identify cell types in single cell genomics. All right, that's it already. I realize I've been quite fast. We would have some time, but I'm finished. Sorry?
Good timing, 1 hour.
So just as a summary, the question I wanted to talk about is how do you explore high-dimensional data to identify order in this high-dimensional data set? And I showed you a couple of dimensionality reduction techniques to visualize your high-dimensional data and that allow you to identify a small set of macroscopic observables to describe your data. I showed you a couple of approaches for clustering to identify discrete order in high-dimensional data. And I think one important point that I want to make is the methods to use to work with high-dimensional data depend on the problem at hand. So really depends on what you're looking for, and often it's just trying out a lot of things to see what makes sense to understand your data. And with this, I'm at the end, and there's some references in the back which I will upload with the slides. And with this, yes, I'm happy to take questions.
Okay, perfect. Thanks a lot, Fabian. Thanks for taking the time, and especially since data people like you are in such high demand these days and very busy. So it's great that you took the time to show us some of your stuff. And so I think what we usually do is that we hang around a little bit, on Zoom. And then if you have any questions, you just stay online and ask your questions. Yeah, and other than that, so then see you all next week. And there's already a question in the chat. How computational?
I realized that there were questions in the chat during the lecture, and I didn't see the chat. So
yes, you can't see that very well if you're sharing the screen. Yes, but I think they were answered already by other people. So now the latest question is, how computationally intensive is UMAP?
That's a good question, and it's actually, that's another advantage of UMAP. It's pretty fast. you don't need a lot of memory. It scales very well with the dimension of your data and also the amount of data points. So I think it's I mean, just like, it's one of the fastest things around at the moment.
I mean, if you look at the typical data sets that you're looking at, where you have, I mean, 15,000 dimensions, and then maybe a few 100,000 or 10,000 of samples.
Yeah, you end up
in a few seconds.
Yes, on my machine, like with the largest data sets I have, which are of the order that you described, it takes about 30 seconds maybe to compute the UMAP, which is much faster. As for instance, there's T-SNE. I don't know whether you know about it, but this was used a lot before, and this is much slower usually. And actually, there's I didn't try this out, but there's also GPU of implementations of UMAP nowadays, and this again, you can nicely parallelize it, and so this should be much faster again.
Mm.
You're welcome.
Great, thanks. So, are there any other questions?
I have a question. It's from UMAP. So when we, in all the examples of UMAP, we saw approximating a high dimensional data. Isn't there any parameter in the algorithm that quantifies the acceptability of the approximation, whether the approximation is how much is it acceptable or not? Because, or will it approximate any high dimensional data? But there didn't seem to be any parameter that quantified or helped us to understand if the result of the UMAP approximation is good or not, obviously concerning the problem at hand.
I'm not fully sure whether I completely understand the question, but here are a few thoughts on this. So the approximation in UMAP is that you approximate that your approximation is that the data is uniformly distributed on the manifold, right? And the question is, and maybe the question is this a good approximation or is it not a good approximation?
Yes.
The problem is that we hardly know the real metric of the underlying space. So if you, for instance, think of these examples with pictures, right? What is a distance between two pictures? What is this metric? I think this is you can't actually, like, unless you have a good microscopic model, which in this case we don't have, it's not really possible to define a good metric, and therefore to judge whether the approximation is a good one or not, it's not.
Yes, I mean the way that you do it is there's no right rigorous way of doing that, and that's why people like Fabian exist, who have experience and have consistency checks, and that with some experience can check whether a UMAP makes sense or not, or whether it's so. One of the standard problems is that a UMAP can be dominated by noise, by technical artifacts, for example, and then you need to have some insights into data and some experience that tell you whether what you see actually represents something that's real in the data, or whether that's something just an artifact. So that's basically one of the jobs that bioinformaticians or data scientists do, because there's just no rigorous way where you just push a button and it tells you whether what you've done is right or not. So it needs some experience and some insights and understanding of the data and the for example, the biology that is underlying the data.
So typically, would the typical method would be to use multiple reduction methods and see which one is giving us an order which is much more perceptible in our plots, figures.
I mean, you also have to know the strengths and weaknesses of each method. For example, what Fabian told about previous before t-SNE, that had a tendency to perform to give you clusters of data points. Yeah, it was not very good at representing global structures of data sets. Yeah, and once you know that, you can interpret what you see, and then you can be careful in interpreting basically clusters as real physical or biological objects. Yeah, UMAPs, the UMAP, what you've seen now, that overcame this limitation, and that it's basically also represented the global structure of data quite well. But I guess also in UMAP you can have artifacts, yeah, especially if your data has noise, yeah, technical noise. You can have very weird artifacts. Yeah, so you need to treat the data before you can give it to UMAP in typical instances. Yeah, so for example, you have to log transform data very often. If your data spans huge orders of magnitude in values, yeah, then sometimes you have to log transform it for not the very few very large data points to dominate everything. And so you have to treat the data in a way to make it, so to say, digestible for these methods.
Okay, thank
but
you.
then maybe, I mean, the other part of the question was in a way parameters, and I didn't talk about parameters at all. And there are a couple of parameters in the algorithm. And I skipped a lot of details, but the most important parameter is the number of neighbors you look at in your, when you construct a graph. If you take the first neighbor, right, you kind of, what you do is you estimate your metric very locally. So this is very fine, but it's also very noisy. So you often will estimate the wrong metric. If you increase the number of neighbors in there, you will get a more robust estimate of the local metric, but you will miss some fine structure of the data. And maybe actually there's one example that I can show from here, where if you look at, I don't know whether you can see this here. This is a maybe you can you see this, or is this too small?
Sorry, I don't see anything.
You're not sharing the screen?
Sorry, I don't share the screen, so you can't see anything. Let me show this example. Kind of, if you, that's maybe actually a good example for your question, where kind of, okay, here we have a circle with dots, and they are like the distance between the dots is not always the same, right? And so actually, if we take just this example and we say, okay, here we know the Euclidean metric, we can think of how well does the approximation now, or how does UMAP behave if in this case, right? And one thing that you can see here is that if I increase the number of neighbors, that this will usually lead to a, actually not. So you can see that sometimes you manage to, for some, there is kind of an optimum for looking at the number of neighbors, which allows you to actually capture the circular topology, the hole in the middle, right? But if you go too big, you kind of lose start to lose the information about the order of these dots, of these measurements, right? They start to get more and more noisy. If you look at less neighbors, you get nice ordered lines, but you start to miss things where you have these gaps, and you start to create clusters where there shouldn't be clusters. So this is the most important parameter of the method. I'd say there is, like, if you the UMAP homepage, from the UMAP algorithm, they feature a nice section of what parameters do you have for UMAP, and what influence do they have. but it is generally relatively robust also compared to other methods. If you go for typical, like it just, but this is just empirical kind of, if you go with about 15 to 30 neighbors or something, you usually derive good representations of the topology of your data if you have enough data points. So it's relatively robust to the choice of parameters. You don't have to play around with it too much.
Thank you.
Okay, perfect. Very nice. Do we have any other questions?
Yeah, there's a question from Kai Müller from the chat, which I will read, which is, he asked, for high dimensional data, how does UMAP determine the manifold dimension? I don't think that it does. And I don't think that it does. And this might be related to that it only, in the UMAP algorithm, what you do is when you construct this, the graph representation from the intersecting spheres, you only look at the one simplices. So you only construct a graph and you don't look at higher order simplices. I'm not sure whether this could be used to say something about the dimensionality of your manifold. I'm unsure there. But UMAP by default, it doesn't tell you the dimensionality.
Okay. Any other questions? We have a lot of questions today. Okay, so if there are no more questions, yeah, then let's end the meeting. Yeah, thanks all for joining, and thanks again to Fabian.
Yeah, thank you for having me. Yeah, thanks.
Okay, perfect. Yeah, see you all next week. Bye.